All right, Science 30. So last little video here in our genetics portion of this unit here. For the most part, we are going to talk about antibiotic resistance. And so antibiotics are things that kill bacterial infections. And so you can have antibiotic resistance occur through mutations, which is what we just finished talking about, by having some sort of a mutation randomly occur in that bacterial organism that ends up making it resistant or mutates it in some way that it doesn't get affected by the antibiotic you're giving it to kill it. Natural selection, right? The survival of the fittest. If you have organisms in a bacterial colony that are resistant a little bit more to the antibacterial than the rest of the species, the population, then they will survive a little longer. And they'll maybe survive long enough to reproduce, and then you'll have more of these antibiotic resistant bacteria present, and then you'll have to, hit them with a harder regime of antibiotics to kill them. This is why they say even if your symptoms stop when you have some sort of an infection, do not stop taking the antibiotics until the entire course is finished. So if they said take one pill a day for 14 days, then you do that for the full 14 days, even if by day 7 you don't feel anything anymore. Even if you feel fine, you need to make sure you wipe out even the toughest of them. Otherwise, two things can happen. The bacterial infection could come back, and this time stronger because you didn't kill all the tough ones. Or you could now have enough oomph inside of your immune system to fight that infection, but you might spread now the antibiotic resistant versions of that bacteria to people around you, and then you're passing on that antibiotic resistant strain. So you need to make sure you take your antibiotics through their full course. And the other way bacteria can be resistant to antibiotics is by sort of passing on their DNA fragments or by passing on entire rings of their genetic information. So really briefly here, we won't jump into too much detail, but if there is a mutation that allows a bacteria to survive, it will pass it on, and then the rest of the population will eventually have that mutation, which makes it advantageous because it won't be harmed by the antibiotic that is being focused on it. Bacteria also can change their genetic information by sort of trading information. So transformation is the process where two bacteria can actually swap info. They can, like at the park and you're trading your Rattata Pokemon card for a buddy's Pidgey and a juice box, right? So you're swapping out information, and sometimes that information ends up being beneficial. And so that's another way that you could gain antibiotic resistance as a bacteria, by receiving antibiotic resistant information. Plasmids are these little circular pieces of DNA, and sometimes in that circular piece of DNA there's extra information which could lead to antibiotic resistance, and then they could transfer those plasmids to a different bacteria and then pass on the antibiotic resistance and antibiotic resistant genetics that way. And so plasmids and transforming DNA are relatively the same for our context, and so we're just saying that bacteria can swap beneficial information back and forth, which might allow them to be better suited to protect themselves against antibiotics, which is good for the organism, but bad for humans. So that's it for this short video series here. Moving on after this, we'll start talking about genetics in terms of pedigrees and different traits. So we'll talk about like Punnett squares and how we pass on heritable traits, dominant recessive alleles, things like that. Okay, thanks guys.